Ever wondered about the magic behind things that glow? You know, the brilliant flash of a highlighter under a blacklight, or the gentle, lingering light of a glow-in-the-dark star? Well, it all comes down to a pretty fascinating story. A story about a single molecule's journey into light. So let's dive in and see how it all works. So this is the big question, isn't it? It's really a tale of two different glows. One is instant and fleeting, and the other is slow and just lasts. They seem like total opposites, but believe it or not, they actually start in the exact same way. On the left, you've got that instant glow, like you see in these fluorescently stained cells. It's absolutely brilliant, but it vanishes the second you turn off the light source. Then on the right, you have the lasting glow, the kind that holds onto its light long after the energy source is gone. We're going to break down exactly what's happening at the molecular level to create these two totally different effects.
Okay, but before we can get into the difference between these glows, we have to start with the fundamentals. I mean, how does matter emit light in the first place?
Now this diagram is basically our roadmap. When light hits matter, it can either scatter or get absorbed. We're going to follow that absorption path. This leads us to a whole family of phenomena called luminescence. And the two specific types we're talking about today, fluorescence and phosphorescence, they fall under a category called photoluminescence, which really just means light that's created by other light.
So yeah, luminescence is the big umbrella term. It's any process where a substance gets all electronically excited and then releases that energy as photons, particles of light. This is the scientific foundation for everything we're about to get into.
All right, let's make this personal. We're going to follow the story of a single molecule as it gets energized and then figures out how to release that energy back into the world as light.
Okay, this diagram might look a little complex, but trust me, it's our key to understanding everything. It's called a Jablonski diagram. Just think of it like a ladder. The higher up you go, the more energy there is. The bottom rungs are the ground state, where our molecule is just happy and stable. All those lines higher up, those are the excited states it can jump to.
So the journey starts at the bottom, in that ground state, S0. Our molecule's just hanging out, minding its own business. Then, bam, a photon of light smacks into it. This is absorption. In an unbelievably fast process, we're talking femtoseconds, the molecule absorbs that energy and just leaps up to an excited state, like S1. Now it's buzzing with all this extra energy, and it has to get rid of it.
And this brings us to the first and most direct way back to stability. That super fast glow we call fluorescence. Let's look at the diagram again. After losing just a tiny bit of energy by vibrating, no light involved, it just kind of shakes it off, the molecule takes the most direct route home. It drops straight from its lowest excited state, S1, right back down to the ground state, S0. And that drop releases a photon of light. This whole direct path, that is fluorescence.
So let's define it. Fluorescence is light that gets released from what's called a singlet excited state. It's just a specific high-energy arrangement of the molecule's electrons. But the key takeaway here is that it's fast, and it only happens as long as you're hitting it with that initial energy. You turn the black light off, the glow is gone. Simple as that.
And how fast are we talking? Well, the average lifetime of this excited state is around 10 nanoseconds. That's 10 times billions of a second. It's so fast it's practically instantaneous, which is why we just see it as an instant flash of light.
But what if the molecule doesn't take the direct path home? What if there's a detour? Well, this is the plot twist that leads to the slow burn of phosphorescence.
Let's go back to our map. The molecule gets excited up to S1, just like before, but instead of dropping straight down, it takes a sideways step. This move is called intersystem crossing. It slips into a totally different kind of excited state called the triplet state, or T1. From there, it eventually falls back to the ground state and emits a photon. This much longer roundabout journey is phosphorescence.
Now this detour is special. In the quantum world, this jump from a singlet to a triplet state is forbidden. That doesn't mean it's impossible, just that it's not very likely to happen. But when it does, the molecule gets kind of trapped in this triplet state. It's a lower energy state than S1, but it's a very difficult place to escape from quickly.
And this trapped nature is what defines phosphorescence. It's a process where the molecule holds onto that absorbed energy for a much longer time before finally letting it go as light. And that is exactly why your glow-in-the-dark stars keep shining long after you turn off the lights.
So just how long is this delay? The lifetime of that triplet state can be anywhere from microseconds to 10 seconds or even minutes. Now compare that to the nanoseconds of fluorescence.
This massive difference in timing is the entire reason for the two different types of glow.
Okay, so we followed the molecule's journey, but this isn't just a tiny physics story. This stuff has huge implications, so let's see how these glows are actually put to work in our world.
This graph shows a really critical idea. Notice how the peak of the absorption curve is at a shorter wavelength, that's higher energy, than the peak of the emission curve. That energy gap between the light that goes in and the light that comes out is called the Stokes shift, and it's a direct result of that little bit of energy loss from vibration we mentioned earlier.
And this shows you exactly why scientists care so much about the Stokes shift. If the shift is small, the light you're using to excite the molecule and the light it's glowing are too similar in color, making it really hard to even see the glow. But with a large Stokes shift, the colors are really well separated, which makes the fluorescent signal pop. It's crystal clear, and that is vital for things like medical imaging and biological research.
What's really cool is scientists can even control which path a molecule takes. Check out this data for naphthalene. On its own, it's mostly fluorescent. But look what happens when you add a heavy atom like iodine to it. The fluorescence yield just plummets to almost zero, and the phosphorescence yield skyrockets. This heavy atom effect actually encourages that forbidden intersystem crossing, effectively steering the molecule right down the phosphorescent path. And these principles are not just lab curiosities. They are the workhorses of modern science. We're talking about DNA analysis, studying how proteins fold, visualizing the inner life of our cells with fluorescent microscopes. These glowing molecules let us see the invisible machinery of life.
So we've gone from the journey of a single molecule, climbing and falling through these energy levels, all the way to this, a breathtaking view of the inner structures of living cells. It's a perfect example of how understanding a fundamental piece of physics can give us a powerful new window into our world.
And that leaves us with a final glowing question. What other invisible processes are out there waiting in the dark that we could bring to light, if we could just find the right way to make them glow?